most of the prokaryotes is going to be cyanobacteria or the green algae.
Hello.
Because you know how I roll, and then Wednesday, I think I'll do eukaryotic algae on Wednesday, and then I've got a plant and fungus lecture. I'll do that later. Might go a little bit out of order from what's on there, but there's some cyanobacteria. I think I took that picture. So they're supposed to like, well, let's talk about viruses for a little bit and other cyanobacteria. So, do you remember viruses? Did you talk about viruses in 130? Maybe? Depends on your professor. Yeah, it depends on everyone gets to it. So just a quick reminder of the structure of viruses. They're not cells, not quite considered life, but they do have genomic information in DNA or RNA form, and they can evolve. So they have some qualities of life, but not all of them. And their genomic info, their DNA, or it could be RNA, is surrounded by a protein coat, and all these little hexagons here are individual viral particles. So they are teeny tiny. And the thing with viruses is they cannot reproduce by themselves. You can't throw viruses on a plain petri dish and have them grow. They've got to get inside of a cell in order to grow. So I think they're making flu vaccines differently now, but years ago they used to ask you if you were allergic to eggs before you got a flu vaccine, because they would grow flu vaccine inside of a chicken egg, because the egg yolk is basically a giant cell. And they can make a bunch of viral particles to get the pieces out to make the vaccines with. So anyway, viruses can't replicate without infecting another cell of some kind. And all species have viruses that infect them, and most viruses are kind of specific to what species they'll infect. There's some that cross the species barrier, like rabies, crosses the species barrier, but some are very specific. Like if you have a cold, you're probably not going to give that to your dog, and definitely not your fish or your houseplant. So most viruses are very specific. And then they're also specific to the type of tissue that they infect as well. So if you get a cold or flu, that's your upper respiratory area. It's not going to be your reproductive organs, for example. So anyway, but all species, animal, plant, fish, insects, algae, bacteria, they've all got viruses that infect them. And just as kind of a freshwater limnological kind of point I wanted to make is that some human pathogens can survive in water and even groundwater. And polio is an example of, I think polio can be found in groundwater. And hepatitis A can be transmitted just through water as well. So that's fun. Alright, that's half of what I know about viruses and aquatic ecosystems.
Isn't that part of the reason why they always tell you to like not drink standing water? Because it's probably built up of viruses and pathogens.
Yeah, for sure bacteria would build up in standing water, and viruses could. Also, I don't know if they would be the kind that would infect humans or not. If someone that was infected had pooped in that puddle, then yes. But I don't know that like hepatitis A is necessarily just lying around. It might be more of a contaminated with sewage kind of thing. All right. The other half of things I know about viruses is, so kind of the reason I'm mentioning them in this class, not just the human pathogen part, but viruses are probably really important in controlling populations of microbes like bacteria and archaea. And let's see, in a couple of weeks, Gruba's going to lecture about nutrient cycles, and you're going to talk a lot about the nitrogen cycle, and you'll mention a bunch of different bacteria that do a bunch of different transformations. And so microbes are really important in keeping those nutrient cycles going. Also the decomposition that microbes do, it's all part of nutrient cycling. So whatever affects bacteria can affect nutrient cycles. And so therefore viruses may also be important in biogeochemical cycles or nutrient cycles, because if they're affecting the size of bacterial populations, that can affect how the nutrients are cycling. And viruses are just generally understudied. So when we think about like food webs, we know about the algae that get eaten by little critters, and those little critters get eaten by fish and maybe insects, and those all get eaten by bigger fish, and so we know a lot about that. But we don't know as much about the bacteria and what eats them. Like, we know a little bit about that, but how viruses affect all those bacteria, that's something that we really don't know very much about. And let's see, this is a paper from a long time ago, a planktonic viral loop. So we've got some prokaryotes in here, and grazing animals, so those would be eukaryotic primary producers as well, which also get infected. Like, all of these organisms could get infected by viruses, and like how everything transfers among all those groups can be affected by viruses, but we don't really know very much like how all that works. So, if you want to go into a field where no one knows anything,
From scratch.
yeah. So that's everything you know about viruses. And I want to talk a little bit about the archaeans. Remember hearing about archaeans in Bio 130? So they're prokaryotes like bacteria, but they're biochemically unique from bacteria, so they're in their own domain. Our three domains: bacteria, archaea, and the eukaryotes. And they, for a long time, we just called them bacteria because they look like bacteria under the microscope, but since we've developed some more molecular tools, we've learned that this group is biochemically unique, and that's why they've been separated out. Bacteria as a group is much larger and has a lot more species, but archaeans are still fairly common, just kind of in general, like they're just around. And they can dominate in extreme environments like the Grand Prismatic Spring in Yellowstone National Park, which that steam coming off, it's really, 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 really hot water. And if you put a fish in there, it would get cooked. But you can find archaea communities living in that hot water. They've adapted to it. So you can find them in some crazy places. But I guess the most common thing that we would interact with as freshwater ecologists are the methanogens, which are, they're considered chemoautotrophs. So as opposed to photoautotrophs, or chemoautotrophs, you remember what autotrophs are?
They make their own food.
Yeah. So auto, self, troph, feeding, so they make their own food. Photoautotrophs, photo means light, so they're photosynthesizing. Chemoautotrophs, they use energy in chemical bonds and break those bonds up to get energy and then make their own food, which is not the same as eating. Like, we eat food to get energy from the food that the food has. We don't make our own food. Yeah.
Does the methane in their name give hint as to what their chemical?
They burp out
Oh, okay.
methane.
It's a
So
byproduct.
they're,
It's
yeah.
not what they use.
Yeah. So, yeah, we call them methanogens because they release methane, and they're common in anoxic soils. So soils that are waterlogged, they tend to run out of oxygen pretty quickly, and so these critters might live in there and burp out methane, which is a greenhouse gas and colorless and odorless and flammable. So you could go out in like a swampy area and like with your lighter, people do this, and like mess up the soil a little bit and try to get a gas bubble to pop up and then light it and it'll go foop. So fun with science. So that's where we might hear about archaeans. And that's everything I know about archaeans. There's not much. And on to bacteria. And so this slide will be some general things about bacteria, and then we'll get into my dear friends, the cyanobacteria. And bacteria are found everywhere in aquatic systems. They live on things and in things and in the water, and they're just all over the place. And they are metabolically diverse. So we'll talk about my photosynthetic bacteria friends. You've got heterotrophs that consume organic particles in some way. You've got other weird things that I don't quite understand either. But there's bacteria have been around for a few billion years, so they've had a lot of time to evolve some weird metabolic pathways. We're used to just thinking about photoautotrophs and heterotrophs because that's plants and animals, but they, like the chemoautotroph thing, they also do that, and there's a couple other things too. Like I mentioned, bacteria are critical in biogeochemical cycles or the nutrient cycles. And they do some transformations. I mentioned the nitrogen cycle. You'll hear words like nitrification and denitrification. And there's different species of bacteria that do that in aquatic and terrestrial systems. Also as decomposers, I think I mentioned that also, taking dead organisms and converting their organic nutrients back to inorganic forms that autotrophs can then take up. So break down dead things to give plants fertilizer, essentially. Very important in food webs. So a lot of critters eat bacteria. Grubau's going to talk about shredders that are insects that like to eat dead leaves that have fallen into lakes and streams, and they're eating these leaves, but they're not getting most of their nutrition from the leaf. They're getting it from all the microbes growing on the outside of the leaf. I call it the peanut butter and cracker situation, where the cracker is the leaf and it's by itself is kind of like whatever, but the peanut butter has all the good nutrients and calories, which is smeared on top. So,
Call it the hummus Etruscan.
hummus Etruscan, yes. Update that one. Yeah, in plankton, bacteria get eaten. I'll talk about some algae that do a little snacking on bacteria in the next lecture. But a lot of things just eat bacteria. A few human pathogens can survive in aquatic systems. So again, usually thinking about like if there's a sewage problem and people that have diseases that can be excreted in feces or urine can get passed on to someone in the water, or who's gotten some water in their mouth, or that kind of thing. You heard about Giardia. You know Giardia, it's not bacteria, but Giardia is a single-celled, not bacteria though, parasite, and it lives in the intestines of various animals, and if they've been infected and are pooping in the water and you drink water that have their poop in it, you can get one of those and get giardiasis, which causes some explosive pooping. It's unpleasant, but I'll talk more about that again. That was one that I thought of, but there's gotta be a bunch of bacteria. E. coli can be pathogenic, and you've got to drink water. So. And just in general, another area, kind of a lot of unknown things, species diversity in bacteria. Not really known. We've estimated that less than 1% of all the species that are out there have had names put on them, which is not much.
Kind of sounds like we just don't really worry about any of the bacteria until it starts becoming a human problem.
Yeah.
It's like E. coli and now COVID. Because there's probably no telling how long it's actually been out there, but then it started infecting the world population.
Right. Yep. Exactly. So, yeah, and the smaller things get, the less that we know about them also. So, small bacteria and even smaller viruses.
It's harder to see.
Yep, it's harder to understand what's going on with them, and so we know less about those. Absolutely. And the other challenge with getting a handle on the diversity of bacteria is that a lot of them look very similar under the microscope, and we need to get at molecular information to get names on these things. And there may be a poster or two with Dr. Kemp and a research student, and basically, like, they take a sample and send off their sample, and someone spits back a bunch of DNA sequences and try to figure out if you've got new species there or what species you do have, and it still seems like kind of a hot mess, honestly, trying to figure out bacterial diversity. And there's just some drawings of what they look like. So, that's almost everything you know about bacteria. But not counting cyanobacteria. Questions? On to the good stuff, cyanobacteria. These actually have a lot more morphological diversity, so they're a little bit more fun to look at. But cyanobacteria, also known as blue-green algae. But since cyanobacteria are bacteria, I guess we generally don't want to call them algae because we want to reserve the term algae for the eukaryotic stuff. The eukaryotic photosynthesizers like diatoms and green algae that we looked at in the lab the last couple weeks. And they are like any oxygen producing photosynthetic bacterium is considered cyanobacteria. And they were the first photosynthesizers on the planet. They put oxygen into the atmosphere. They continue to supply a good chunk of our oxygen. Have you thanked a cyanobacterium? I don't think you have. You gotta think about that.
Doesn't most of our oxygen come from algae and not trees?
That's right.
So we've been lied to since elementary biology.
That means we
People just
can
don't
do
know.
more.
That does not mean we can't. There's lots of other reasons. I mean, there's still a lot. I think the estimates vary, but I want to say it's like 30% terrestrial, and then the rest is algae of various sorts. Alright, so there is a lot of variability in size and morphology. So there's some little teeny tiny cells, and then these are a lot bigger. We've got filaments, we've got weird colonies. We've got, this is a weird colony, out of focus, but looks like cookies on a cookie sheet, another weird filament. So they're very distinctive. Well, not all of them. Most of them are fairly distinctive, we'll say that. We can put names on them. So they're, I think I talked about this in ecomethods. I get confused between my ecomethods lecture and this lecture, so I don't think I've mentioned this yet, but algae in general, we would just, like, when we describe their morphology, we want to talk about if they're unicellular, just single cells, doing their thing, like our individual diatoms that we saw last week. Colonies, bunches of cells that live together, but there's no, like, differentiation. The cells all do the same thing. And then the filamentous ones, the ones that make long strands. So we have a lot of cool looking cyanobacteria. And cyanobacteria, important in the carbon cycle. So this goes to what we just talked about here. Important primary producers, important oxygen producers. So 30 to 50% of global primary productivity just comes out of cyanobacteria. And so if terrestrial is 30-ish %, don't quote me, then the rest 40-ish % is from all the other algae and seaweeds. And these are, a lot of that's in the ocean because the ocean's so big, and these are some little tiny dudes that are often seen in the ocean. And so that's great, giving us all of this oxygen that we're enjoying, using and taking out carbon dioxide too. And the thing with cyanobacteria that kind of messes with ecological stuff in all ecosystems, all places where you find them, I guess, is they're not really a good food source. Like typically you think photosynthesizer, that's good food for somebody. And cyanobacteria is not a very good food source. So they're kind of like apple cores and celery string. Like, don't really want to eat that stuff. Peach bits. So you can, if you get a lot of cyanobacteria growing, there's generally not going to be anything that'll eat that to keep it down. And so that's why when we hear about algal blooms, it's often cyanobacteria, because there's nothing that's really going to eat them. Decomposers, however, will eat cyanobacteria after they die and have lived their little lives. And that will also potentially become an issue. If you've got too many bacteria cells, that means you've got too many that are going to die, and once they die, they get broken down by decomposers. Decomposers use up oxygen, and that can be bad for fish and junk like that. Okay. Mm-hmm.
So what keeps their numbers down? Is it just the space being taken by other species that?
Around here it's really temperature. Like in the wintertime, they're not, and they don't like cold very much, and so they.
So environmental factors.
Yeah, pretty much. And if they're in a stream, it would be like if they get blown out. Okay, so cyanobacteria, they often outcompete other algae in certain conditions. And one of these would be high nutrients, and we'll talk about why that is in a little bit. And also, they like warm temperatures, so late summer, early fall is when they tend to be at their maximum. And so if you were to Google harmful algal bloom right now, you might get a lot of stuff. Also, they can do pretty well under low light conditions, which ultimately means they can grow a little bit more in deeper water, potentially. So that can contribute to just having more stuff, more cyanobacteria. Like I mentioned, they tend to kind of max out midsummer to early fall. And then when it gets cooler and darker, even though it's darker, they like darker, but when it gets cooler, the other algae tend to do better. And so Pacer Pond, we saw green algae, that was the broccoli looking stuff, but there was also some cyanobacteria in there. If we were to look again in January, probably wouldn't have as much stuff. There might be still some of that green broccoli looking stuff, but the cyanobacteria really wouldn't be there. Then there would be other things that we would see. I think compared to my Canvas file, I moved some slides around, so I don't know if I did it or not. Prepare yourself. Okay. All right. Okay, a little bit more about cyanobacteria and nutrient cycles. So this species, or this genus, is Delicospermum. Used to be called Anabina, but they change names all the time. This is in Pacer Pond. It's super common all over the world. And there's some weird looking cells in here that I want to talk about. And these smaller round cells that are the same size as normal cells, but they have a different kind of color to it, different sort of texture going on. Those are cells called heterocytes, and they used to be called heterocysts, but the cyanobacteria people decided cyst was too much of a eukaryotic kind of word, and so they wanted something that would be suitable for prokaryotes, and cyte just means cell, and so they have heterocyte, just meaning a different cell that looks different. And heterocytes, what happens there is nitrogen fixation. Y'all remember what nitrogen fixation is? Start with atmospheric nitrogen, N2. How much of the atmosphere is nitrogen? 78%. There's a lot. And N2 is a triple bond, and so it doesn't like to be broken apart. So as each inhalation that we take, we're bringing in 78% of those molecules are nitrogen gas, and then we just breathe them right out. They don't react with our cells at all. And nitrogen fixation is what some bacteria can do to take that nitrogen gas and convert it to, you can see that, ammonium, which then a photosynthesizer could use. They could take that up and use that for nutrition. So cyanobacteria, they can, if they have heterocytes, they can make their own nitrogen. They don't have to get it from their environment.
Wait, did you just say heterocytes make their own nitrogen?
So heterocytes are the cells that make nitrogen. And so if a cyanobacterium has heterocytes, there's certain species that have them and certain ones don't, then they can just make their own nitrogen.
Fixes it from N2 to.
Yeah. And so nitrogen is a very common limiting nutrient. There's often, it's often what we put in fertilizer because the plants would grow more if only they had more nitrogen. And so this is also the case in aquatic situations, and so if they can make their own nitrogen, they can also use that to outcompete the other algae as well. So phosphorus is there's two big nutrients in aquatic ecosystems, nitrogen and phosphorus. Those are the two that are most often limiting. And again, it's if we just had more of one of these, we could grow more, do our thing. And cyanobacteria that cannot like make phosphorus like they can nitrogen, but they can hog it. They can basically take nitrogen and store it in their cells for later use. And they can store a lot, like 100 to 200 times more than they actually need.
Do they store it in like the useful form ammonia or like ammonia?
So it's phosphorus, so
Oh, phosphorus, sorry.
they make polyphosphate bodies, so they have like little granules basically in their cells. And that's all I know about that. Things that happen inside the cell are like hard for my brain. That are outside of the cell. Imagine if you could carry around 200 times more donuts than you actually needed, but just have them with you all the time in case you just wanted a donut. That's what they're doing. Instead of something that they actually need. Don't need. Okay, so main point, cyanobacteria, they've got an edge over other algae because a lot of them can make their own nitrogen, and if they're, I'll give you an example of how luxury consumption of phosphorus would be useful. But if they're in a situation where there's extra phosphorus lying around, they can just kind of hoard it and use it when they need it later. Right. Okay. So, all this talk about cyanobacteria because they are big bloom formers, algal blooms, where you get a whole bunch of algal growth, generally in plankton, and it can cause problems, as we will discuss. And this is microcystis, which is another common bloom former. That Delicospermum from the slide, they can also bloom. Microcystis can also bloom, and it's because of that making nitrogen, storing phosphorus, and I forgot to mention this, but all those heterocytes, they're making all this nitrogen basically, that also leaks out of them. So organisms are leaky, anything in the water is like they take in nutrients in some way, and then nutrients can just leak out of their tissues. It can be in waste products, but they're also leaking out nutrients. So that can, like the whole bloom thing, if you've got nitrogen fixers, they can be feeding themselves, but also others around them a little bit. Another reason we see nasty blooms of cyanobacteria is some of them are extra buoyant. They like to float to the top, and when you get everybody like floating toward the top, you get this thick layer of goo.
Which, does that block out the light for any algae?
It could, yeah, if it's, like the more biomass you have toward the top, and this is a microcystis bloom where you've got a lot kind of floating up, it can block light for, like plants that might be growing out of the soil, they'd be in the dark. Absolutely. And they're buoyant with structures called gas vesicles. And so this microcystis, we're looking at it in the microscope with the light shining through it, and it looks brown, and that's because of the nitrogen vesicles that it has, just little nitrogen gas bubbles in there. And so it's kind of just an artifact of light going through that. If you could shine the light on it and have it reflect off of the cells, it would be this color. So it's actually kind of an artificial color, but it's like dark green. So that those gas vesicles help them float, and they can actually control how much gas is in those vesicles, which allows a phenomenon called diel migration to occur. Do you know what diel means? So, diel migration is referring to, they can go like up during the day to photosynthesize and then go down at night to get nutrients. And so they go through a migration over a 24-hour cycle. And which you can see diel migrations in the water column of other small critters as well. There's some little animals, little zooplankton that live in the water, and they might hide more toward the bottom during the day to hide from visual predators like fish, and then come up toward the top at night to feed and hide out from their predator. But the cyanobacteria, they can do that also, which is kind of wild. They're also good. They're often the algae forming blooms because, like I mentioned, they're not a good food source. Like nothing is really eating them. And if you've ever been around a gnarly cyanobacteria bloom, you'll smell it because it's nasty. It's a very unique odor. And I've only really smelled it one time, and I've got pictures to show you from that. But just having all of that stuff growing in the water, they can ooze out chemical compounds, make the water kind of stinky. If you use that water as a water source, it's hard to clean that up. It can make fish taste bad. It's gross.
So is that like the typical swampy smell?
No. That's a good smell. Yeah, this is very like kind of pungent and almost industrial. Like it's
Not the dog food factory.
no,
smelly though.
nope. It's very unique. I can't even describe it. I don't know how to describe it. It feels like it. And as we will talk about further in a little bit, some species of cyanobacteria or some strains of certain species, they will make toxins, and so they're just an algal bloom on its own can turn into a toxic algal bloom. Right. So, long story short, usually algae blooms are cyanobacteria because of all of those reasons. I'm gonna skip this one for now and come back to it. This is the algal bloom that I wanted to show you. This is where I smelled the smell. So this is a genus called Aphanizomnon, and they're filaments, and these are heterocytes. And the filaments like to stick to each other and make these little clumps that you can actually see if they get big enough. And this was in Iowa when I took the diatom class at Iowa Lakeside Lab in 1998. And all this junk floating on top, it looked like someone took a dump truck full of grass clippings and just dumped it in there. And it was just like really thick and nasty. And it smelled bad. And so that's a slide. I had a slide, like a slide, and I scanned it. That's how old that is. That's kind of a top-down view, kind of hard to tell, but all the light color is the Aphanizomnon. Closer to shore, this is kind of out of focus, but it was sort of blowing into shore and kind of concentrating it. And so where cells would die, their pigment would leak out, and they actually have water-soluble pigment. And so this blue, it looked like latex paint, which was really weird. And then next to shore, all that turquoise color is all of that pigment. And there's up on shore with a bunch of pigment kind of washing in, and then just chunks. And it smelled really, really gross. Just like really gross. And we were there just looking for diatoms, and we did not see any diatoms at all. it was all this defenazone. Yeah, let's talk about harmful algal blooms. Since we're talking about the stinky stuff. So, HABs, harmful algal bloom. And some blooms can be toxic, but you never know it unless you test it. So you can have a big, gnarly, stinky, gross cyanobacteria bloom, but you won't know it's toxic unless you test it. Species that make toxins don't make them all the time, and we're still kind of sorting out why they produce toxins, like when, under which conditions they do, and which ones they wouldn't. But for safety's sake, you should always assume that if you see that kind of pea soup look, that you have a toxic algal bloom, especially if it's a place where you wanted to go swimming or wanted to let your dogs go swimming. Don't let your dogs swim in that because they'll drink the water, and if it's toxic, it could kill them. Like, dogs have died from cyanobacteria blooms. And so freshwater, all of our harmful algal blooms are typically cyanobacteria, and it's only a few species, like a few types that will produce toxins, but they're often the ones that make these big nasty blooms. A lot of them are not toxic. If you're in the ocean, just to mention it, dinoflagellates, which is a group that we'll talk about them on Wednesday. If you've heard of red tide, they make red tide. Have you heard of red tide? It's another nutrient problem thing. There's a few cyanobacteria in the ocean that might bloom, and there's also some toxic diatoms in the ocean that you don't see in the freshwater. So that's fun. And toxins can cause problems if they're ingested, which, like, you wouldn't want to go swimming because it's easy to accidentally swallow water if you're swimming. If you let pets in there, dogs are notorious for drinking water as they're swimming. They can irritate your skin if you just touch it. And all that stink that I was smelling, if there were a bunch of toxins in that bloom, they could aerosolize. And then if you inhale it, that can cause respiratory problems for people with either someone who was exposed to a lot of it, or if they have sensitive respiratory issues. And a few years ago in Florida, they had some red tide that was doing the same thing. Like, people with respiratory issues were like warned to not go outside and like walk on the beach because it would be unpleasant for them. So there's that bit, and I didn't say anything about how I need a slide for this. It was just a fun story. I'll tell you about that later. Another big problem with algal blooms is that, like I said, the cells live, die, and then they get decomposed. Once they're decomposed, decomposers use up oxygen, and so you can get fish kills in ponds and lakes because those decomposers of the big algal blooms, no matter what kind of algae it is, they use up the oxygen, especially at night. In the day, all that algae would be photosynthesizing, producing oxygen, but at night, they're using it up, and their decomposers are using up oxygen. Plants use oxygen too, we just don't think about it because they give off more than they use, but they use it at night. Or they use it all the time. You can measurably notice that they use it at night because they aren't making any. So plants are burning up the sugar that they make in mitochondria just like we are, which uses up oxygen. Okay. Oh, this book. Coffee table book about algae. And there's a chapter with cyanobacteria. Some cool pictures in it. These are good pictures. Here's some more fun ones. Look at these little guys, they're so cute. Is that cystis. This is a book. Do you know what a book is? It's made of paper.
I don't know, they kinda don't make those anymore.
I know.
But they force us to buy one every
More cyanobacteria.
I know, I spent too much money on them than I need.
This was probably the first organism seen in a microscope by Anton van Leeuwenhoek. 350 years ago. Like 350 years ago, September 17th or something like it's, or September 5th. It's anyway, there's a bunch. Cyanobacteria pictures in here. So, this keeps on going, but that's all I had for today. So we'll do the other algae.